Hello, thank you for joining me. So before I do my case study, I just decided to go through some of the theories and principles with regards to the CAR-T structure and the way that the various designs have been made, the design of the biological entity, the biological living cells, living T cells, in order to be as effective as possible in the clinic, in the patient, and reduce the potential side effects, negative effects that are associated with this type of modality. So this is just kind of like giving all the first principles so that when I get to doing the case study, I don't have to do a long study and I don't have to do long presentations and long documents. So there's just a few things that I just wanted to cover and clear up, because I just made some assumptions in my previous presentation of prior knowledge, and we all have to start somewhere. So these chimeric antigen receptor T cells, they are applicable to CD8 cytotoxic T cells, which are naturally occurring anyway. Then CD8 plus T cells are cytotoxic in nature, and I've gone through how we do that in previous presentations. But these T cells are also engineered for the CD4 plus helper T cells as well. So as well as performing the helper functions, if you like, that the CD4 plus T cells do, have evolved to do, they can be engineered. The CD4 plus cells, T cells, can be engineered to express this chimeric antigen receptor and therefore have a cytotoxic effect. And so these CD4 plus CAR T cells can directly kill tumor cells, and they're not just merely supporting or helping the CD8 plus cells. They do help the CD8 plus cells, so they have almost like a dual function, if you like. But when they are engineered with this chimeric antigen receptor, the CD4 plus T cells, they can acquire that cytotoxic function, and therefore they have that ability to perform mediated killing, if you like, via this perforin and granzyme B enzymes. And so the other thing to note, though, is that the cytotoxicity, if you like, or the cytolytic activity, I don't know if that's a word, is less potent than the CD8 plus CAR T cells. But nevertheless, you want to get optimized, and you want to have, as much as a potent drug as possible with minimal side effects for something like tumor or cancer, solid tumors and cancers. So I just wanted to make that point that yes, we're talking about CD8 plus cytotoxic T cells, because those are the most cytotoxic T cells. They've evolved to be that way, and CD4 plus T cells are also cytotoxic when they have been engineered to express that chimeric antigen receptor. So what have I got here? Yeah, and as I said, they also, CD4 plus also have a helper effect. They're T helper cells, and they can also produce the relevant cytokines that are necessary for the formation of other T cells. And these are really what we're looking for is the central memory T cells and the stem cell like memory subsets, or the stem cells like memory T cells. That's what we're looking to to get when we have our distribution of final product, that is a distribution of a number of cells. We're looking to guide the get it to the right kind of I don't know, proportions or distributions where we have good effector like functions, as well as memory like and stem cell like characteristics. So we want to have that, and that is supported through CD4 plus T cells, which have that helper functionality. So as well as having the chimeric antigen receptors, you also have other receptors that are engineered into these T cells. So these T cells are super in terms of like Superman as opposed to your ordinary man, in the sense that they not only do they have these chimeric antigen receptors that are designed to kill cancer cells, but they also have other synthetic receptors that are designed to overcome the tumor microenvironment that, you know, that the tumor microenvironment is what causes the tumors to evade the natural immune system. But what we have done is we have re-engineered the T cells so that they don't respond as they typically or as they have evolved to, but we flipped the switch. So basically this TGF beta, which is secreted by the tumor microenvironment, overly secreted, overexpressed, is normally responsible for switching off and downgrading the T cells, so that they are no longer killing the tumor cells, but they're just basically bystanders. And so what has happened is that what these synthetic receptors do is that they have been, this TGF beta receptor binds to an activating region, an activating domain, and now instead of being turned off by the microenvironment, the T cells are activated. So it's these synthetic receptors, and there are synthetic strategies to improve particularly tumors that are difficult to treat, CXCR2. This is trafficking receptors that traffic these chemotaxins or chemokines, and they are responsible for guiding the T cells to where the tumor microenvironments are, so that they can be more effective. And this IL-7 receptor, these are responsible for guiding the formation of certain central memory or stem-like cell memory T cells that have better persistence. So in development, we're taking advantage of where there are gaps, and we're looking to improve the overall efficacy of these T cell therapies. CD16 is also a receptor, synthetic receptor, that is normally produced or naturally found on antigen presenting cells. Is it antigen? Well, no, sorry, naturally found on the immune cells, so natural killer cells and other immune cells. And it's required by antibodies. So antibodies will coat a particular cell. This is opsonization. And then these immune cells will bind with the antibody. So you have your cell, you have your antibody, and you have the immune cell complex. And then that will trigger the ADCC, the antibody-dependent cellular cytotoxicity. Well, what's happened is that these T cells now have been engineered to do that. And so as well as your natural killer cells or your other immune cells, you also have these T cells that can now destroy tumor cells. So it's a very multi-strategic approach of ensuring that you get as much killing of these tumor cells as possible. The other types of antigens, antigen binding types of CAR T cells are where you have this dual antigen recognition CAR T cells. So there are two types. There's the dual CARs and then there's the tandem CARs. So the dual CARs is where you have T cells express two separate chimeric antigen receptors, and they're basically for two separate antigens. So you have antigen A and antigen B. In this particular case in clinical trials, it's CD19 and it's CD22. And in order to get activation, you have to have both of those binding to occur. So you need to have binding of CD19 to one of the CARs and binding of CD22 to the other CAR. And what you get is one of the CARs, say antigen A, which could be CD19, is the co-stimulatory domain, and the other CAR, antigen B, which could be CD22, is the CD3 signaling domain. And as you know, for T cell full activation, you need to have the signal, the main signal, and the co-stimulatory domain. And so for your T cells, this is a much better way of improving the specificity. And then the next one, and I think that would be to make sure to reduce the cytotoxic effect. Well, not cytotoxic, that's the word, the cytokine release syndrome, and to reduce the negative side effects that would be, and to make it much more specific, to really target specific cancer or tumor cells. And then the other one is the tandem CARs, and this is where you have a single CAR that contains two single chain variable fragment part of the antibody part, and it's all in the same linear chain. So you have a single chain SCFV, the variable part, for A, antigen A, and you also get it for antigen B, the SCF variable for antigen B, and it's joined together by a hinge, and then that is, sorry, it's joined together by a linker. So each SCFV for A and SCFV for B is joined by a linker, and then that's attached to your hinge, which is also attached to your transmembrane domain and your intracellular domains. So what you have is you binding of either of the antigens, CD19 or CD22, can trigger activation unless you engineer it such that you can have it or both of them together. So it depends on how it's been engineered. So there are various different, I think that the purpose of this slide is to show there are multiple, and this doesn't include all of them, multiple strategies that a developer can take and that are in development, and I have not included all of them, but there are multiple strategies that developers can take in order to design their T-cell therapy. And these synthetic receptors are, this is just some of them. It's just to make that point. So I've got this, and I've called this the basic CAR structure, because in this particular example and in everything that I'm going to be talking about from now on, I'll be talking about a single SCFV, so a single antigen binding site, not the dual ones that I've mentioned previously. But this is what I'm going to be talking about. What I'll be talking about in this particular presentation is this whole structure and how it's all been designed to have a specific, to optimize the therapeutic ability, if you like. And every part of that particular, of the T cell has been engineered in order to carry out a specific function in the most optimum way possible. It's really quite beautiful if you think about it, the way that they've thought through everything. So for example, you have your antigen binding domain, and this is the single chain variable region of the well, it's derived from an antibody, that part and that specific part, and it's the variable region, not the heavy part or the constant region of that antibody. And then you have your hinge and the spacer, and that is a very important part. There's certain properties and characteristics for optimal optimal behavior, if you like, or optimal characteristics. And then you have your transmembrane domain, which is important for anchoring the whole of the CAR from the extracellular and the intracellular roles. And then you have your co-stimulatory domain. This is the CD28 or the 4-1BB, which is the second signal, and it's required for full T cell activation. And then you have your signaling domain, and that's the CD3 zeta, and that's important, and that's what gets activated when you get binding. So in order for full T cell activation, you have to have signal 1 and signal 2. But this whole structure, if you like, this whole CAR structure, chimeric antigen receptor structure, it has been, every part of it has been designed in order to get optimal optimal effect. And there are different kinds of designs. So there are different designs or things that people have looked at in experiments where they've looked at, say, for example, the length of the hinge, how that affects the ability for the antigen binding site to operate and to bind strongly. And the transmembrane domain, what properties, what are important for that, to be able to, what are we looking at? For example, dimerization is important in terms of having a stable, stable stable activation or stable signal activation. All of these things are really important. The co-stimulatory domain, the type of stimulation that we're looking at, is it strong stimulation, strong effector-like functions, which we normally get with CD28, or is it more measured, which we can get with 4-1BB? And there are the two different types of CAR T cells depend upon or have different, they have different co-stimulatory domains, and you choose them based upon the type of cancer the patient's presenting with and what you want in the long term or initially. And then you have your CD3 zeta, which is the typical signaling domain, but that in itself has been it's been re-engineered. I think I'll be talking about it in later presentations, but it has been re-engineered in order to control the activation and ensure that you don't get this cytokine release syndrome or excessive cytokine storms. So this is sort of my basics, which I'll be using. I'll be coming back to this slide on a number of occasions. So let's talk about the antigen binding. So first of all, we'll just talk about the target antigens and just get that out of the way. Here are a list of all the target antigens. I've given the names and where they are normally expressed and where they are usually overexpressed in cancers. So the reason why we're targeting them is because of the fact that they are always expressed in a particular cancer, or they are always overexpressed in a particular cancer. So HER2 is the human epidermal growth factor receptor 2. And it's overexpressed in breast cancers, ovarian cancers, non-small cell lung cancers. So it's key in solid tumours, but it is low-level expression in epithelial tissues such as breast, lungs, and gastrointestinal tract. So we have to optimize and really think about activity of your T cells so that you get the right activation and you get the right therapeutic effect on cancers, and you limit the effect on non-cancerous cells that also have the antigens present. So I guess you can look at this on in your own time. CD19 and CD22, I think I've already discussed, as I've also discussed BCMA, B cell maturation antigens. These are CD19 and B cell maturation antigens. I think they're already marketed medicinal products. CD22 I definitely know is in clinical trials, and I definitely know there's dual antigen receptors. Mesothelin, this is overexpressed in certain cancers. Pancreatic cancer, ovarian cancers are really difficult to treat with standard clinical practice, and therefore just thinking about making because they are overexpressed in these particular cancers, but they are also expressed in mesothelial cells, thinking about designing the product so that the drug targets the tumor cells and has a much more activated in the tumor cells where they're overexpressed, where there's a higher density, and where they are also expressed on normal cells, but because the density is going to be not so high, you don't get that activation. Mesothelin or mesothelin, they are GPI anchored cell surface glycoprotein. So basically, GPI anchor is a glycolipid that tethers proteins to the outer leaflets of the cell membrane. So that's another. Go and read a biology book on that, because I had to. So it's basically surface presentation and tumor restricted expression makes it valuable for makes it a valuable target, if you like, for CAR T cells and other antibody-based therapies, and that's the reason why we're looking at it. It's a mesothelin is a solid tumor marker. And we can also use CD22 as a backup target on B cells, like the CD19. And GPC3, this is only really expressed in embryonic tissues, in fetal proteins, but it is re-expressed in liver cancers, and that's why we're targeting, we're using it as an antigen that we're targeting, designing our chimeric antigen receptors to target. So I'm just going to talk about the single one. I'm not going to talk about dual, but the same thing applies to both of them, and that is that the single chain variable region of the antibody consists of a variable heavy region, or the heavy region variable part, and the light region variable part, and that's it. It doesn't consist of the constant region, and that's really important to note. I mean, the variable region is what does the binding. Historically, the first single chain variable fragments were derived from murine, the mouse monoclonal antibodies, because mice are very easy to immunize against human tumor antigens, and they generate strong specific antibodies. And then you would need to humanize those antibodies, because obviously you don't want them to be recognized as foreign I don't know, particles by the patient's immune systems, and you don't want that. You don't want that immune rejection, because these are their T cells. You don't want the T cells to be rejected by their body. So you need to humanize those fragments, antibody fragments, as much as possible, such as modifying the antibody sequence to make it look more human while keeping that antigen binding specificity. You know, that's kind of traditional, if you like, way of looking at it. But for more products that are in development, they are either fully human or from specific libraries, or they're synthetic. So you know that those are the direction that we're going. We're becoming more effective in terms of reducing potential for rejection, reducing the potential for side effects, and increasing specificity and increasing the effectiveness of the drug. um I want to talk about antigen density because it's just something that I think we should know. It's first principles, and just making assumptions that people understand things when maybe they do, maybe they don't. So tumor cells are usually, there are some small tumor cells, small sized tumor cells, but they're usually quite large. Yeah. And normally more than one, numerous T cells attack the tumor cells. So it's not just one T cell that's attacking the one tumor cell. And these T cells contain multiple receptors. Yeah. So wherever you have a tumor cell that has a high antigen density, number one, that means that there is a lot more chance for the chimeric antigen receptors to grab hold of. So the chances of activation improves. And also you're going to get much more stronger activation, because there's more CART that's engaged, more of the receptors are engaged, chimeric antigen receptors are engaged. So the T cell itself that contains numerous chimeric antigen receptor is going to have that strong activation. So if you have a highly dense, high antigen density, you're going to get, and that's the aim, is to get stronger activation. And as we know, activation is going to drive certain things. You're going to drive the cytokine to be released, and this IL-2, which is released for T cell growth, which is necessary for effector functions, T cell effector functions that increase the tumor killing ability, higher, much more effective in terms of tumor killing. You get that initially with your activation, on activation. So, and you also get IFN gamma, that's interferon gamma, and tumor necrosis factor alpha as well. So that's what you really want to get. Now, where you have low antigen density, it's harder for the chimeric antigen receptors to find enough binding sites. So you get weaker, basically no activation. So if you have optimized your T cell, so that you have more than one, if you like, well, if you have optimized your T cells and to go for high antigen binding or high antigen density, you're more likely to get activation. But for the cells, normal cells that do not normally, well, they are not tumor cells, but they do have the antigen, but the antigens are not overly expressed, they're not overexpressed, you're not really going to get that activation. You'll get very weak activation, which is what we want, because we don't want to, we don't want to get to target and destroy normal cells. So, so as I said, the cancer cell is large and the antigen density is very high, yeah, or there's a high local concentration of your CAR T cells. That can be the other point. The other thing that I said, as I was talking about synthetic, other synthetic receptors, you know, that if you've got these synthetic receptors, they can target, they're attracted to various signals that are produced by the tumor microenvironment. So if the T cells migrate towards and swarm around these tumor cells, especially if they're producing chemoattractant signals, so the CXCL9 or CXCL10, you get that activation. And this is good because it increases the chances of rapid killing, and you get this faster destruction of big or resistant tumor cells. There is a problem in that you can also get, if you're attracting the T cells to a small area, you can get this local cytokine storms. You know, if you get too many T cells that are firing off at once, and you can potentially get tissue damage, but these can be managed, if you like, through it can be managed. You have to take a risk-benefit approach, It can also be neutral because it depends on the context. Some tumors need a heavy T cell to break through their particular defenses. And as I said, normal tissues might express low amounts of the target antigen, but they might also have, if you've got high affinity CARs, these are CARs that are strong, that you don't require a lot of them to get activation, so they're high affinity, you've got strong binding. You could, if you have designed those types of CARs, where you have high affinity CARs, they might still attack normal tissue, and this is what we refer to as on target off tumor toxicity. So you really, well, what is actually in clinical trials at the moment, or something that I've looked at, is where we're looking at low affinity CARs, where the signal is lower, has been reduced, specifically designed and reduced to help where there are antigens, lower antigen densities, but you don't want to get that activation, so you reduce your off-tumor risks. So there are two things happening at the same time. You're having your direct activation through antigen binding, but if you have too few CAR T cells, it's hard to get that compounding effect. But if you have too many CAR T cells and you have a high antigen density, you do have this higher risk of cytokine release syndromes. And you just basically, all the clinical trials have to balance the CAR T doses and monitor for all of these reactions. So I talked in the previous slide about affinity, where I'm talking about the binding of one CAR, the receptor, to the binding antigen, and the strength of that is determines the affinity. So if it's strongly bound, then you've got a high affinity. If it's not strongly bound, then you get a lower affinity. But as I said previously, that the T cells contain more than one receptors, if you didn't realize, and therefore you're getting multiple bindings if the antigen is overexpressed, if you've got a high antigen density. So you kind of have a cumulative effect if you have a reduced affinity. So you can have a strong high affinity CAR, or you can have a reduced affinity CAR where you have multiple bonding that increases the ability of that T cell to become activated. So Avidity is looking at how many CARs on the T cell bind and the strength of that, of the affinity of each of those CARs, whereas the affinity is looking at the single strength. And what you want is you want to optimize it such that if you have a lower affinity but you have overly expressed antigens, the avidity is going to be higher and therefore you'll get more effective killing. But if you have low antigen density, you're not really going to get that activation, and therefore you will reduce the effects of your on-target off-tumor effects. You'll reduce the negative effects or the likelihood that you're going to get that T cell activation. So avidity is something that we have to look at. Yeah, and here's a slide. Here's a slide that just talks about what I've just said, which is avidity is important because even if each individual CAR antigen bond is moderate, that's the medium affinity. If you've got lots of them engaged at once, the total avidity can be strong. And the higher the avidity, the more stable the synapse between the T cells and the tumor, and the stronger the activation and the more reliable the killing. And avidity helps compensate if a single molecule binding isn't super strong, yeah. And for HER2, chimeric antigen receptors might have a lower affinity to reduce off-tumor toxicity, but because the tumor cells have a high HER2 density, many CARs can still engage at once, giving a high avidity, and you get that strong activation where it's needed. Right, now this one is about the hinge and the spacer. So as I said, I've talked about the antigen binding single chain variable fragment, and now we've moved on to the next part, which is still outside of the cell, the T cell, and it's the hinge or the spacer part. And this is usually derived from CD8 alpha. You can get IgG1 constant region that's used as a hinge or spacer, or you can have IgG4 FC constant. Now, there are going to be issues with using the IgG1, i.e. you have to ensure that you don't get off-target immune activation, because, and I'll talk about that in some next slides. But through trial and error, and most of the work that I've seen, CD8 alpha is what's usually used. However, for certain cancers, you know, there are various formulations out there that uses different hinge spacers because of the length and the type of the resistance of the cancer due to various cellular characteristics. So the hinge or the spacer is really important because, you know, it's the length and the flexibility and the fact that hopefully it is of human origin to reduce the ability to reject. The tonicity, this is just through trial and error. My understanding is from everything that I've read. You know, you want to have low tonicity, you want to have low sort of spontaneous activation. All of these things are dependent upon the hinge length, and I will be going through these. But the common sources and the most used are the CD8 alpha. So it's the flexible spacer, is the flexible part of the whole construct, if you like, the whole receptor construction, if you like, that is outside of the T cell itself, and it connects to the antigen, the SCFV, and the transmembrane domain, which is the molecule that anchors the whole structure into the cell membrane. Yeah, so the hinge and the spacer region, it provides flexibility. The hinge gives the CAR molecule enough of sort of space or room to be able to move, so that the FCFV can properly sort of orient itself and bind to the target, even if the target antigen is, you know, positioned awkwardly, like if it's close to the cell membrane, or maybe it might be sticking out far into some dense tissue. You know, having that hinge flexibility allows the receptor to be able to get to the antigen. The reach is important because, as I said, some antigens are buried close to the cell surfaces, and as I said, some are sticking out. So if you have a long, more flexible hinge, that allows for better access to antigens that would otherwise be hard to grab. But there are some issues with that, in that you can increase your chances of off-tumor, on-target effects. And then the hinge also influences the immunological synapse formation. So the quality of that synapse it determines how strong, or it affects how strong and sustained the activation is. So all of these parts are really important with regards to activation, and a well-designed hinge helps to form a tight functional synapse. Tonic signaling is important. Poorly designed hinges, you can end up with unwanted activations even when there's no target. So this is tonic signaling, and that can lead to T-cell exhaustion. So tonic signaling is where you get spontaneous activation of your T-cells in the absence of its target antigen. So it's almost like a false alarm. And that can lead to T-cell exhaustion or uncontrolled cytokine release, and basically shorten the persistence of your T-cells, and also have very little or very poor tumor killing ability. So, you know, the hinge and the spacer region, even though, well, I don't know, it's not something that you might think about, it's very important in terms of the overall ability for that cart to what's the word I'm looking for, function optionally and to perform. And you wouldn't think so, you'd just be focusing on the well, I would have been focusing on the antigen itself, but every part of this receptor has been specifically optimized in order to get the best possible outcome. I've got that it can affect immunogenicity because of the fact that, you know, I was talking about the CD8 alpha is what's normally used, but sometimes there are some where the construct consists of the IgGF, is it IgG1 FC or IgG4 FC, yeah, and some hinge regions can be recognized as foreign by the immune system, but usually they are human derived to reduce that kind of rejection. So here's kind of what I think I've already summarized this, where CD8 alpha is short and flexible. It's very common, and it's used for it's used mostly for most of the formulations, or not formulations not the right word, but the way that the design of the receptor. And then you have this IgG1 FC, which is longer, but it's rigid, and it's needed for distance or membrane close antigens, but you can get the problem with off-target effect. And the same for IgG4 FC. So I said here feature it's modified to reduce FcR gamma binding, and it's safer than IgG1. And this FcR gamma is something I will talk about in the next slide, but it relates to antibody binding. That so antibodies can bind antibodies bind to immune cells to get that antibody dependent cellular cytotoxicity. But if the immune cells bind to this FcR gamma binding, yeah, then you get unwanted cytotoxicity, unwanted activation of your T cells, and unwanted killing of your T cells. So those those regions have to be mutated or modified in some way to prevent that from happening. And there's a very famous clinical trial, which I will talk about, where we've learned this lesson. So the hinge length, these are short, medium, and long. The CD8 alpha, this is the medium one, which is the best. It's a little bit like Goldilocks. The short one is too short, and you may struggle to reach the antigens that are close to the tumor cell surface, and you get poor engagement. And the long ones, yes, you get easy engagement, but you do have a higher risk of engaging normal cells with low antigen density, although that can be you can make adjustments for that, but still the long hinge IgG1 FC can improve tumor killing, but there is that problem with off tumor toxicity risks. And as I said, the FC gamma receptor has to be modified because you don't want those negative effects. So I think I've already said that, and you can have a look and have a read of that in your own time. So I want to talk about the FC binding, because if you use IgG1 or IgG4, the FC fragment crystallizable region, you do have to modify it. So the FC region is the tail of the antibody, and what happens is it binds to FC receptors, that's FC gamma Rs, on immune cells like macrophages, natural killer cells, or dendritic cells. And this binding in normal situation helps trigger an immune response like phagocytosis or ADCC. Yeah, so that's what normally happens. But CARs use this well, they don't use the use of the CD8 alpha. I haven't looked very much into that, but the potential to use the FC region may still be present, and if it is present and if it is seen by the immune system, then you have a problem. Because if the FC gamma receptors bind, so the FC gamma receptors are present on macrophages, natural killer cells, and dendritic cells, if they bind to the FC gamma, to the FC region on the actual T cells, you get off-target activation of innate immune cells. So the macrophages and the natural killer cells, they will become activated. You'll get unwanted inflammation, and then you'll get non-specific T cell activation, which is dangerous, because you can get these cytokine storms and cytokine release syndrome. And then you get the depletion of your CAR T cells by phagocytosis. So your macrophages will eat up your T cells, and you'll end up reducing the amount. So it's obviously not what we want. But you must modify or remove FC binding features to avoid accidental immune engagement. And this is something that is standard practice in the design of these types of CAR T cells. So this is kind of what I've been saying, where these are the engineering that's involved. This is what you do. You would remove the FC, the residual FC parts, so that there's no chance of FC gamma receptor binding, or you can mutate the FC-like regions so that you abolish that recognition, so it's not going to recognize it. And if there are any murine antibody parts that can be seen as foreign, you humanize them, so you make sure that you're not going to get that problem with rejection. Yeah, and that's just basically what I've been saying. The FC region must be modified or silenced to prevent accidental binding to the FC gamma receptors. And this avoids non-specific activation and protects the CAR T cells and protects the patient as well. So you want to basically silence that FC region so that you don't get that binding to the FC gamma receptors. And this leads us very nicely into the TGN 1412 trial disaster. Basically, this product, this clinical trial product, was a very harsh learning curve for it occurred in the United Kingdom. So this TGN 1412 is a super agonist, and it's anti-CD28 monoclonal antibody. So basically it's required to, you know, that CD28 is required for activate. It has to be activated in order to get your activation of your full activation of your T cells. Yeah. And sometimes you don't get that activation for various reasons. It could be because of the fact that the CTLA4 effectively outcompetes the CD28, or you just don't get full activation. Whatever the reason is, you're not getting that full activation. And so this super agonist was designed to make that happen and to have a hyper activated T cells. Yeah. And they did perform preclinical animal models, and it did look safe, but it wasn't carefully screened for FC gamma receptor interaction. So remember, this FC gamma receptor is on the natural killer cells, it's on the macrophages and dendritic cells, and it wasn't screened for that. Yeah. So the species that they did use wasn't specific enough. It wasn't similar. The FC gamma receptor was not similar to humans in the species that was selected. So in these first in-human trials, which was in 2006 in London, the UK, phase 1, 6 healthy volunteers were dosed. I believe there were 6, and within minutes all of them suffered life-threatening cytokine storms, and they were hospitalized in intensive care, and this became one of the most famous examples of first-in-human trial disaster. Right, so what was the problem? First of all was this unexpected activation of the immune cells. And this was due to this FC receptor binding, yeah, and this species mismatch between the humans and the animal models that was used in the preclinical studies. So, as I've said, the FC part of the antibody binds to the FC gamma receptor on the immune cells, yeah, but we don't want that to, obviously don't want that to happen. So basically these antibodies were given to the patient and they cross-linked with the FC receptors, and then you had this massive cytokine release where you had this IL-2, which is quite a strong cytokine and produces more T cells, and then interferon gamma and tumor necrosis factor alpha. And the problem was that it wasn't seen in the animal models because the FC receptor expression was different. And so this dangerous reaction wasn't seen. And basically, there was a lot of outcomes that resulted from that. But basically, the FC binding must be carefully, well, I meant to delete carefully controlled. Well, it must be silenced, especially in therapeutic antibody fragments, but also in CARs as well. It has to be silenced, not carefully controlled. It must be mutated, or the gene must be mutated, or the residues must be, the peptide residues will be unrecognizable, such so that you don't get that FC binding to the immune cells. What have I got in my notes? Yeah. So this is now standard practice, really. So all CAR designs will mutate the FC regions if any of them are present, and you'll test for unwanted FC gamma receptor binding in multiple species before you go into human trials. And also, you have this staggered approach where you don't just apply it to all patients at the same time, but you apply it to one at a time. Actually, I've got another slide on that. Yeah, here it is. So basically, new regulations were created in the EU and the United Kingdom as it was at that time. And that is to, number one, to start for immune therapies, to start at very low doses, micro doses, to stagger the patients, so don't dose everyone at once, and also to have better predictive in vitro assays. Also, something that I noted, well, this is something, and that's to really consider the use of healthy volunteers for immunostimulatory agents. Is those kinds of patients, healthy volunteers are not the best humans, if you like. It's better to go straight into patients. It's just not good practice now. And this was in 2006, so almost over 20 years ago. So it's not something that's done. But this is the outcome that arose from that. And there's an EU guideline, and this is the guideline and strategies to identify and mitigate risks for first-in-human and early clinical trial with investigational medicinal products. Right. So that's the FC region, the hinge and spacer region. The next part is the transmembrane domain, and that's really crucial for activation. So again, go back to our diagram, and the transmembrane domain is the part that is within the actual transmembrane itself, the cell membrane of the T cell. It's a key building block. It has a functional role, not just in keeping the whole thing together on the outside of the cell as well as on the inside, but in this dimerization role that helps to stabilize activation. So it's important it has an important structural and functional role. It anchors the CAR in the membrane, as I said, and it acts like a bridge. And again, that's usually CD8 alpha, CD28, and CD3 zeta in its structure and construction. So the transmembrane domain is, it affects how stable the CAR is in the membrane. It has to be compatible with both the extracellular and the intracellular domain, and the common sources are usually CD8 alpha. So the transmembrane domain helps to control the stability, the expression, and the signaling characteristics of your chimeric antigen receptor. It's, as I said, it's in that hydrophobic domain. So it will consist of a short stretch of hydrophobic amino acids, yeah, and it anchors the CAR in the cell membrane, so that it's so that some of the outside part, it sticks out into the extracellular region, and the inside part is in the T cell region, the intracellular signaling machinery. So it acts like a bridge between the two, and it's derived usually from CD8 alpha, can very rarely from CD3 zeta or CD28. Basically, what you really look at, well, let's go through the list. So stability. So the transmembrane domain affects how stable the whole CAR is in the membrane itself. The CD8 alpha transmembrane domains are the ones that are mostly used because they're reliable, and they form these dimers. And these dimers are basically it consists of two entire chimeric antigen receptors molecules on the same T cells that pair up into dimers across the membrane. And this is good because it stabilizes the CAR structure without accidentally activating it. Not all types of dimerization are good, yeah. So sometimes you have these transmembrane domains are derived from CD28 or certain IgG4 spaces, and you end up getting random clustering of CARs, and that can sometimes lead to tonic signaling, yeah, which can cause the T cells to activate without there being any tumor antigens present, which you don't want, because that's going to lead to exhaustion. So CD8 alpha is what's usually preferred because it forms just the right amount of dimerization. You don't get random self-clustering, you don't get spontaneous activation, and it stabilizes the whole structure to these two structures and is ready to fire when the antigen actually binds. Yeah, as I said previously, the transmembrane domain has to be compatible with both the extracellular domain and the intracellular domain. Otherwise, you're going to get problems. You're going to get this disruption of the receptor and disruption of the function. It's just not going to work, basically. It has to be compatible with both. And that's why CD8 can be used, sometimes CD28, if it's linked directly to a CD28 co-stimulatory domain. But, you know, it's mainly CD8 alpha. Let's look at the signaling part. So the signaling part really consists of your CD3 zeta and your co-stimulatory. So you need to have both of them. You need to have both the signal 1 that comes from the CD3 zeta and the signal 2 that comes from the co-stimulatory CD28 or 41BB. Yeah. Right. So I think this slide is in the wrong place, but I might as well talk about it since it's here. And the CD3 zeta, which is responsible for the activation signal that occurs when the binding occurs between the antigen and the antigen binding part of the CAR T. The CD3 zeta consists of 3 separate, if you like, regions or sequences of amino acids, and these are called ITAMs. So ITAMs are small sequences of amino acids that are found inside immune cell receptors like T cell receptors and chimeric antigen receptors. They're the CD3 part, yeah. And what they do is they basically responsible for the activation. They kick off the activation, and so they consist of. There are really 3 parts to it, yeah. And they sit inside of the cytoplasm. And when the antigen binds, the tyrosine residue in the item gets phosphorylated. That's just how it triggers the activation, and then that triggers a cascade of activation and signals inside of the T cell. Yeah. So, but it typically, the CD3 zeta typically has 3 ITAM motifs, and remember, each ITAM is a small sequence of amino acids. And as I said, in order for it to be activated, you get this, you have this phosphorylation where a phosphate group is added, and that triggers a cascade of activation signals inside a T cell. What have I got for my notes? Yeah. So for my case study, this is the first part, this is the introduction part. HER2 targeted 1XX CAR integrated into the TRAC locus. So let's break that down. So HER2 is the antigen that's overexpressed in the cancer cells or the tumor cells, which the single chain variable region of the antibody part binds to, yeah. The 1XX, what that refers to, if I remember the ITAMs, so it's basically the three CD3 zeta, which has 3 separate ITAMs, as I said, but 2 of those ITAMs have been knocked out. It's been disabled. So basically, instead of you get 1/3 of the activation. So you reduce the potential for overstimulation. And that's good, because what you want is T cell persistence. You want the type of T cell, the memory phenotype, and it's better with certain solid tumors because you have this less of a cytokine release syndrome risk. So I've also got something about TRAC, and that's because this slide was taken from another presentation, and basically I'm talking about knocking out the T cell receptor alpha constant and knocking in the CAR T cell expression. And you really want to do that because you want to utilize the natural endogenous part of the genome for your chimeric antigen receptor. You want to reduce the potential for graft versus host disease because the T cell receptors, if you're looking at, say, for example, an off-the-shelf product, you might have that issue. So that's just background information. Yeah, so this is the point that I was making, is that the CD3 zeta consists of 3 items, and 2 of them have been mutated so that they're non-functional, and you only have the 1, and basically you have a weaker CD3 signal. But where you have overexpression of an antigen, that makes up for your avidity. So you might have what's the word I'm looking for? You might have a weaker affinity, but a stronger avidity, and this means that you will reduce the off-target effects. The off-tumor effects, so the on-target off-tumor effects, and reduce you have a weaker activation, but it's more balanced activation signal and more preferential in controlling that activation and controlling the potential cytokine storm. think I've already covered that, so there's no need to go through it. But the basic concepts are that if you have this reduced activity, 1/3 of what it should be, you're going to reduce T cell exhaustion, you're going to get better persistence, and you just balance the cytotoxicity. So it's still effective, but it's not overly aggressive, and the risk for cytokine release syndrome is very much reduced. So I don't really need to go through this. Just want to talk about co-stimulators because that's an important part of your T cell structure, if you like. So as I said, that for the full T cell activation, you require CD3 zeta and your co-stimulatory domain, which is the signal 2. And so signal 1 is your antigen specific, and that comes from your ITAMs, and signal 2 is your co-stimulator. And your co-stimulatory domains can be either CD28, which is what it usually is, or 4-1BB, either one or the other. Both of these are co-stimulatory molecules, and they provide the second signal that's needed for full T cell activation, but they both have different activities, if you like. But without this co-stimulation, even if you get T cell binding or chimeric antigen receptor binding to the antigen, the T cells are not activated. They'll become non-responsive, and they may even undergo apoptosis. So you always need that second signal, co-stimulatory signal, to get full activation. So I just want to compare the two, CD28 versus 4-1BB, sorry. So 4-1BB is usually on the natural ligand is the antigen presenting cells or dendritic cells. CD28 tends to have stronger activation, so you're going to get more IL-2 production, and it tends to promote effector T cell functions, so you get that strong killing effect. Whereas 4-1BB is lesser in terms of activation, but more long term. So in the CD28 you have this immediate proliferation and cytokine secretion, whereas the 4-1BB is more about the long-term effects. You want to have long-term survival, you want to have resistance to apoptosis, and maybe your T cell memory, the T memory memory T cells, and your stem cells like formation. Those are what the 4-1BB tends to promote. So this is the co-stimulatory parts of the whole of the CAR T cell structure. So again, this is what we have. The activation speed is fast for CD28 compared to 4-1BB. Your cell expansion and proliferation is more rapid. The downside is you have a shorter persistence compared to 4-1BB, where you have a longer persistence. Your cytokine profile is slightly different, more different. You get more of your IL-2 and interferon, and these are your IL-6 is more associated with cytokine storm. But anyway, your cytokine profile is different. It's more robust, if you like, for CD28 compared to 4-1BB. And as I said, your T cell phenotype is more effector-like in its function, the more of the killer cells, whereas the 4-1BB tend to generate more memory-like cells, and they're different products. So it depends upon the patient, what kind of therapy is dependent on the patient. So, you know, if you need a therapy that acts fast for aggressive, rapidly growing tumours, CD28 might be better. But if you're looking more long-term control, you know, to prevent the relapse in leukemia, say, 4-1BB might be better. Although some groups, I believe, and some clinical trials are combining both to see if they can get the best of both worlds. I've talked about dual co-stimulatory domains in experimental CARs, have I? No, I haven't. I've talked about dual CARs, but I haven't talked about dual co-stimulatory domains in the experimental chimeric antigen receptors. So, you know, this is basically overall CD28 is rapid, it's less durable, but 4-1BB is slower, but it's more longer lasting. And both of these signals, either one or the other, is essential for co-stimulatory, but they differ in how they shape your T cell behavior. Okay, so here is some background information. So basically there are different T cell subtypes, and depending on your manufacturing process, you might want to drive the distribution of your product to have certain types of certain T cell subtypes over others. But here they're the different ones. Your naive cells, these are not activated at all. Then you have your central memory T cells, and then you have your effector memory T cells, and you have your stem-like memory T cells. You really want to get your stem-like or central memory T cells long-term, and they're more desirable because they expand well in vivo, they persist long-term, and they respond to repeated antigen exposure. And if you're using induced pluripotent stem cells, you can direct the differentiation into certain subtypes, those particular subtypes, using certain cytokines like IL-7, IL-15, and notch signals, which I'm not going to talk about in this presentation. So cytokines, they basically control your immune system. Like I think of it like hormones control a certain metabolisms. The cytokine controls the immune system and the immune control and response. So they're produced by immune cells such as T cells, macrophages, dendritic cells, and they regulate your immune activation, inflammation, and healing. And they can act locally or systemically to coordinate your immune responses. And, you know, like I've talked about IL-2 and IL-6 and interferon gamma and tumor necrosis factor alpha. Those are the common ones when we're talking about T cells. So the immune system uses cytokines to tell the immune cells what to do. So your process is activation, and then you get your amplification, so you get that proliferation and expansion of your T cells. And then you also have going on at the same time systemic effects by the cytokines in order to improve that activation, drive that amplification, produce more persistent cells or memory cells or whatever it is. And then after all that, you want that regulation. You want to bring everything back down and calm the system down. You want to produce the memory cells. You want to tone everything down. And the reason you want to do this is because you don't want the body to be activated. You don't want your immune system to be activated all the time, or else you're going to be looking at autoimmunity. So that's kind of what I was trying to put together here. Now, the cytokines have various roles and are released by various T cells. So the CD4 plus cells release some of these cytokines, as does CD8. But they're required for, IL-2 is required for T cell proliferation, activation, and survival. So you get when the cell is highly activated and it's high effector function, it's the IL-2 that's doing the job. IL-6 is implicated in cytokine release syndrome. IFN gamma, this is for macrophage activation. It's important for killing tumors, but excessive levels, you can end up promoting cytokine release syndrome. So here, yeah, all of these are key cytokines in CAR T therapy, and you can look at this in your own time. The same here. These are the same, but I've just got a little bit more information. I was just trying to get it in my head. Now, one of the things that I'm going to talk about in my case study is ICANS, and ICANS stands for immune effector cell associated neurotoxicity syndrome. So this is this neurotoxicity that you can sometimes get as a result of excessive cytokine release, and it can occur after CAR T cell therapy or other immune effector cell therapies. And the other thing that occurs is this cytokine release syndrome, but ICANS specifically refers to neurological. It's a neurological toxicity. So the symptoms will be headaches, confusion, difficulty speaking, seizures, tremors, reduced consciousness, and in severe cases, brain swelling, cerebral edema. So it's believed to be cytokine-induced endothelial activation and blood-brain barrier disruption. And it's triggered by high levels of cytokine. That IL-6 that I referred to earlier is implicated in this, and interferon gamma is also implicated in this. And this is how it's managed. This IL-6 blockers is applicable to or helpful with cytokine release syndrome, but not particularly effective for ICANS. But basically supportive care, anti-seizure medication if necessary, and using steroids. So this is the end of my presentation. I've just got some references. I don't think that I put all of them there. So here are some of the literatures that I've referred to that you might want to look at. Here are some of the books that I read. Here are some of the clinical trials, not all of them. Here are some of the websites for clinical trials. And this is what I will be doing one of my case studies on. However, it's not the only thing. I haven't finished my case study. So this is a very long presentation. I hope you found it to be useful. I'll try and separate it up into different parts. Thank you so much for bearing with me. And this is the first part in the case study, and it's just the background first principles information, so that when I do talk about what the case studies, I don't have to go through and go through and talk about these parts that I've talked about in this presentation. Thank you so much for your time. Bye bye.